The overall goal of this procedure is to retrieve DNA from active microorganisms that have consumed a growth substrate of interest without the prerequisite of laboratory cultivation. This is accomplished by first incubating an environmental sample with a stable isotope labeled substrate of interest under conditions that are similar to those found in the native environment. The second step of the procedure is to extract total DNA from this isotope labeled sample. The third step of the procedure is to subject this DNA to density gradient ultracentrifugation in cesium chloride. The final step of the procedure is to fractionate the density gradient to obtain heavy and light DNA for subsequent molecular characterization. Ultimately, results can be obtained that reveal the identity of active yet uncultured microorganisms involved in the metabolism of a particular substrate through a variety of possible molecular techniques, including fingerprinting, microarray hybridization, clone library analysis, and metagenomics.
Hello, I'm Josh Neufeld from the Department of Biology at the University of Waterloo.
I'm Eric Dunford, a graduate student in the Neufeld Lab.
Today we'll be showing you a procedure for DNA stable isotope probing, or DNA SIP, which has become a widely used technique for linking the identity of unknown and potentially uncultivated microorganisms with their ability to use a growth substrate of interest.
We use this procedure in the Neufeld Lab to study the metabolism of carbon sources in a variety of terrestrial and aquatic environments.
Today we'll be showing you the application of a particular carbon substrate, 13C-glucose, to a soil sample. But we acknowledge that since this method was first developed by Colin Merle's group at the University of Warwick, researchers have diversified the protocol to be applied to a variety of different environments and also using a variety of different labeled isotopes like nitrogen and oxygen. Today we'll be focusing on carbon, but the protocol itself can be applied to different experimental designs.
So let's get started.
Prior to the start of this procedure, prepare all DNA stable isotope probing, or DNA SIP reagents, as described in the written portion of this protocol. DNA SIP begins with sample incubation. Incubation conditions vary widely depending on a variety of sample and substrate characteristics. Once appropriate incubation conditions have been determined empirically, add the appropriate substrate amendment to the environmental sample. You may also choose to include supplemental nutrients to enable microbial growth if these are required based on test incubations. Once the sample incubation has been set up, cap and seal the sample. Then, incubate the environmental samples containing labeled substrate. The example being shown involves the addition of a carbon-13 labeled glucose solution to a soil sample. Next, set up a control incubation to serve as a comparison to confirm apparent labeling of nucleic acid observed in subsequent steps. Prepare this sample under identical conditions except using unlabeled carbon as the substrate. Cap, seal, and incubate the environmental sample with unlabeled substrate under the same conditions. Following the incubation, extract DNA from the microcosms using an extraction protocol that is suitable for the desired downstream applications. Then, quantify the extracted DNA for the following ultracentrifugation steps using an agarose gel and a spectrophotometer. Use vertical or near vertical well rotors for ultracentrifugation to ensure maximum possible separation of light and heavy DNA. The Beckman Coulter VTI 65.2 rotor has 16 wells for holding 5.1 ml quick-seal polyomer tubes. To prepare samples for ultracentrifugation, first calculate the volume of extracted DNA that is required to add 0.5 to 5 micrograms of DNA to the ultracentrifuge tubes, using the previously determined DNA concentrations. Then, determine the exact density of the cesium chloride stock solution using a digital refractometer as suggested in the written protocol. Calculate the volume of gradient buffer DNA solution required to generate an appropriate mixing ratio. Using these calculations, combine the extracted DNA with gradient buffer and 4.8 milliliters of cesium chloride in a sterile, disposable 15 milliliter tube. The resulting volume will be greater than the specified capacity of the ultracentrifuge tubes in order to completely fill the tubes. Finally, mix the solution by inverting the tube 10 times. Variations of this protocol use a fluorophore, such as ethidium bromide within the gradient solution, to enable DNA to be visualized within the tube, altering the density of DNA. When prepared with pure culture DNA, including an ethidium bromide gradient as shown here is particularly useful to provide immediate visual confirmation of gradient formation following each centrifuge run. Steps in using such an approach are described in the written protocol. To set up the ultracentrifugation, first use a bulb and Pasteur pipette to carefully fill the ultracentrifuge tubes with the gradient solutions. Label the tubes on the tube shoulder with a fine permanent marker. Ensure that the tubes are filled exactly to the base of the tube neck, as insufficiently filled tubes have a tendency to burst during ultracentrifugation. Once all the required tubes are filled with the sample solutions, record the precise mass of each tube. Sort tubes as closely matching pairs, and add or remove minute quantities of solution until they are balanced to within 10 milligrams. Keep the solution level as close to the base of the tube necks as possible. Then, seal the tubes using a tube topper according to the manufacturer's instructions. Check that the tubes are sealed properly by inverting them and applying moderate pressure. Weigh the tubes again to check that they are still balanced after sealing. Prior to ultracentrifugation, carefully inspect each rotor well to ensure they are clean and free of any dust and debris that could puncture the tubes during ultracentrifugation. Insert the tubes into the rotor, placing the balanced pairs opposite each other. Record the rotor location of each sample, as the ultracentrifugation process can render marker labels illegible. Carefully seal the rotor wells as indicated by the manufacturer. Once the rotor wells have been sealed, load the rotor into the ultracentrifuge. Close the ultracentrifuge door and apply a vacuum. For the VTI 65.2 rotor, set the rotation speed to 44,100 RPM, the temperature to 20 degrees Celsius, and the ultracentrifugation time to 36 to 40 hours. Ensure the vacuum is on, select maximum acceleration, and turn off the brake to ensure the gradient is not disrupted by deceleration. Immediately following ultracentrifugation, remove the rotor carefully. Be careful not to tilt or bump the rotor to avoid disturbing the gradients within the tubes. Finally, gently remove the tubes for gradient fractionation. DNA can be extracted from the ultracentrifuge tubes using the fractionation technique. To begin, fill a sterile 60 ml syringe with sterile distilled and deionized water containing sufficient bromophenol blue dye to provide a dark blue color. Place the syringe on the loading arm of the syringe pump. Then, attach pump tubing fitted with a 23 gauge 1-inch needle and turn on the pump until some water comes through the end of the needle. Avoid any air bubbles in the water supply, as they will negatively affect the fractionation process. For each sample, prepare 12 sterile 1.5 ml microcentrifuge tubes with labels indicating the sample number and fraction. Next, fix one of the ultracentrifugation tubes to a clamp stand. With a quick and controlled motion, pierce the bottom of the tube along the tube seam using a fresh 23 gauge 1-inch needle and rotate the needle to ensure a uniform hole has been formed. You may find that latex gloves provide better grip on the needle shaft than nitrile gloves. Using the needle that is attached to the pump tubing, pierce the top of the tube on the upper tube shoulder along the seam in a quick and controlled manner. Note that piercing the top of the tube followed by the bottom of the tube can also be done. Also note that a small amount of mineral oil coating the shoulder of the tube can help prevent leaking from an improper seal around the upper needle. With a previously calibrated pump rate, pump the dyed water into the top of the tube to yield 12 425 microliter fractions in 12 minutes. Use a timer to collect the gradient solution in the labeled microcentrifuge tubes. Finally, check the density of all fractions for at least one sample or control gradient using a digital refractometer. Expect values in the range of 1.690 to 1.760 grams per milliliter, with a median density of approximately 1.725 grams per milliliter. To precipitate DNA from all fractions, first add 20 micrograms of linear polyacrylamide as a carrier for precipitation, and mix by inversion. Then, add 2 volumes of PEG solution, and again, mix by inversion. Leave the tubes at room temperature for 2 hours to allow the DNA to precipitate. Following incubation, place the samples in a microcentrifuge with the tubes oriented in the same direction to ensure consistent localization of the pellet. Centrifuge the tubes at 13,000 G for 30 minutes. After centrifugation, retrieve the samples from the microcentrifuge. Then, carefully aspirate and discard the supernatant. A pellet should be visible at this stage with the aid of a bright light source. Wash the pellet with 500 microliters of 70% ethanol. After washing the pellet, centrifuge the samples at 13,000 G for 10 minutes. Following centrifugation, carefully aspirate and discard the supernatant. The pellet will be more visible now, but will dissociate from the tube wall more easily. Allow the pellet to dry at room temperature for 15 minutes. Once pellets have dried, suspend each pellet in 50 microliters of TE buffer. Finally, run 5 microliters of each fraction on an agarose gel to characterize the gradient fractions, using methods discussed in the written protocol. Typical DNA SIP results will demonstrate a separation of labeled and unlabeled DNA in the gradient formed by ultracentrifugation. Ideally, there will be a complete resolution of high molecular weight genetic material, such as carbon-13 and nitrogen-15 from unlabeled material. In particular, unique PCR-based fingerprints associated with fractions 4 through 8 of stable isotope incubated samples, but not with native substrate incubated controls, provide strong evidence linking specific organisms with the metabolism of particular labeled substrate. When performed on DNA from 2 pure cultures, carbon-13 labeled Methylococcus capsulatus strain bath and unlabeled Cynorhizobium mellilati ATCC 1021 labeled and unlabeled genomic DNA separate into respective gradient fractions with differing densities. In this example, heavy isotope labeled DNA can be observed in fractions 4 through 5, whereas unlabeled DNA is found at high concentrations in fractions 9 through 10. PCR amplified DNA from the same fractions were run using denaturing gradient gel electrophoresis, or DGGE, and generated discrete banding patterns corresponding to the 2 organisms included in the gradient. The density of the fractions ranged from approximately 1.580 to 1.759 grams per milliliter, and they are shown in order of decreasing density from left to right. Conversely, the separation of isotopes in environmental sample incubations may be more difficult to interpret. Tundra soils from Resolute Bay, Canada were incubated with either carbon-12 or carbon-13 labeled glucose for a 14-day period at 15 degrees Celsius. The agarose gels of the purified gradient fraction DNA revealed smeared genomic DNA across fractions 7 through 10 for both carbon-12 and carbon-13 incubations. However, when DGGE of 16S ribosomal RNA genes was used to determine the carbon-13 enrichment of biomass from particular microbial taxa, the carbon-12 glucose incubated soil DNA generated similar patterns across all gradient fractions, while the carbon-13 glucose incubated sample generated DGG fingerprints that are uniquely associated with fractions 5 through 8. The conserved bands indicated by the arrows represent a dominant phylotype consistent across all gradient fractions. The phylotype shifts to heavier fractions for DNA obtained from carbon-13 glucose incubated soil. The identity of this particular 16S ribosomal RNA gene can be determined to guide subsequent metagenomic or cultivation-based approaches.
We've just shown you how to perform a DNA stable isotope probing experiment from incubating samples to obtaining labeled DNA for molecular analysis.
When performing this procedure, it's important to consider your experimental design carefully. Be careful to avoid adding too much substrate in order to avoid biasing your experiment towards fast-growing organisms.
You also want to avoid extended incubation so that substrate doesn't dilute to other members of a community through a food web. So you want to use just the right amount of substrate and just sufficient incubation times to allow you to detect a small amount of labeled DNA in heavy fractions with sensitive PCR-based applications.
So that's it. Thanks for watching, and good luck with your experiments.